Greetings all. In the Level 1 video for cellular respiration, we talked about the balanced chemical equation for the oxidation of glucose. In the Level 2 video, we talked about the metabolic pathways that together make up cellular respiration. Here for Level 3, we're going to start just by looking at glycolysis, which is the first of these metabolic pathways. Glucose has a lot of potential energy, but you can leave a bowl of sugar on your kitchen table and not have to worry about it exploding. Now, if your kitchen has a pure oxygen atmosphere and you leave a lighted candle on the table, well, that's different. In the investment phase of glycolysis, we're going to spend the potential energy from 2 molecules of ATP, which is kinda, sorta, like the lighted candle. In the payoff phase of glycolysis, we're going to make 4 molecules of ATP, which is sorta kinda like trying to explain to the arson investigator what happened to your kitchen. As we go through the 10 steps of glycolysis, I'll be ignoring the fact that some of these hydrogen atoms can dissociate as hydrogen ions. I'll just show everything as the uncharged molecules until we get to the end. All of the same pieces are still there, whichever way we look at it, but I think it makes it easier to keep track of everything if we just look at the molecules. I'm also going to show the 6 carbon atoms in glucose like this, individually numbered. The carbon atoms will keep these numbers throughout the rest of our conversation here. And finally, I'm showing glucose and fructose in their straight-chain conformations, because we don't need to worry about fancy scientific words like beta-D-glucopyranose and so on in order to visualize what's happening in each step. In the first step of glycolysis, we're going to add a phosphate group to the number 6 carbon of glucose, making glucose 6-phosphate. The phosphate group was provided by a molecule of ATP, which means that step 1 costs us some energy, and therefore this reaction is the beginning of the investment phase of glycolysis. But the energy we invested means the glucose 6-phosphate has even more potential energy than plain old ordinary glucose. The enzyme that catalyzes this reaction is hexokinase, which you might notice is under the reaction arrow in the middle of the frame. A horrible detail that we'll save for later is that the very same reaction is also catalyzed by another enzyme called glucokinase, but glucokinase is used in different cells under different conditions with different enzyme kinetics and is regulated differently, because sometimes we do that. And I put a running total of all the atoms at the bottom of the frame. The phosphate group from ATP added 1 hydrogen atom, 1 phosphorus atom, and 3 oxygen atoms. In step 2, glucose 6-phosphate is rearranged to become fructose 6-phosphate. This is so we can chop it in half in a couple of steps. If you want to hear some fancy scientific words, it's because glucose is an aldose, while fructose is a ketose, and the chopping in half step only works for a ketose. The enzyme that catalyzes step 2 is glucose 6-phosphate isomerase. In step 3, we're going to spend another molecule of ATP to add a phosphate group onto the number 1 carbon, so now we have fructose 1,6-bisphosphate, bis for 2. The enzyme that catalyzes this step is phosphofructokinase. We're now up to 6 carbon atoms, 14 hydrogen atoms, 2 phosphorus atoms, and 12 oxygen atoms. Fructose 1,6-bisphosphate has a lot of potential energy, and it's just absolutely begging to be chopped in half. Step 4. It's chopping time. The enzyme fructose bisphosphate aldolase, which is often called just plain old aldolase, cuts fructose 1,6-bisphosphate between the number 3 carbon and the number 4 carbon. The hydroxyl group that was on the number 4 carbon becomes an aldehyde group, and the leftover hydrogen atom bonds to the number 3 carbon so that the number 3 carbon has 4 bonds again. The half containing carbons 1, 2, and 3 is dihydroxyacetone phosphate, and the other half, with carbons 4, 5, and 6, is glyceraldehyde 3-phosphate. In step 5, the dihydroxyacetone phosphate is converted into a second molecule of glyceraldehyde 3-phosphate. The enzyme here is triose phosphate isomerase, and it converts dihydroxyacetone phosphate and glyceraldehyde 3-phosphate back and forth. Whichever one you have too much of relative to equilibrium is going to get turned into the other one. In a beaker in a lab, triose phosphate isomerase catalyzes the reaction in either direction, but here in glycolysis, it only goes in this direction, as the G3P keeps going away. It's the raw material for the next step. while DHAP and G3P keep getting made from step 4. Here I've flipped the upper right G3P around the horizontal axis, and then in the lower right G3P I've spun the number 4 carbon around so that both molecules look the same. What had been the number 1 carbon in the glucose molecule is now in the equivalent position as what had been the number 6 carbon in the glucose molecule. Step 6. This is a really important step in glycolysis and a really crowded frame in the video, as we're now entering the payoff phase. Back in step 1 and then again in step 3, we added phosphate groups by transferring the phosphates from ATP. In step 6, we're going to add 2 more phosphate groups, but these new phosphate groups aren't coming from ATP, they're just plain old ordinary phosphates floating around in the cytosol. And I'm showing them here as our old friend phosphoric acid, H3PO4. The enzyme that catalyzes step 6 is glyceraldehyde 3-phosphate dehydrogenase, and the dehydrogenase part of the name tells you that we're also pulling off some high-energy electrons and putting them as hydrogens onto our old friend NAD+ to make NADH plus H+. We've now made 2 molecules of 1,3-bisphosphoglyceric acid, and we have a total of 4 high-energy phosphate groups. These are the high-energy phosphate groups that we're going to use to make the 4 molecules of ATP as we complete the payoff phase of glycolysis. Step 7, we're going to start making some ATP molecules. The enzyme is phosphoglycerate kinase, and it takes the high-energy phosphate group off the number 3 carbon and the number 4 carbon to make 3-phosphoglyceric acid and our first 2 molecules of brand new ATP. This process has a fancy scientific name, substrate-level phosphorylation, which is when we make a new ATP molecule by transferring a high-energy phosphate group from some other molecule onto ADP. We can do that here because the bond between the phosphate group and the rest of the 1,3-bisphosphoglycerate molecule is of even higher energy than the phosphoanhydride bond in ATP. Step 8. The enzyme phosphoglycerate mutase rearranges 3-PG to make it 2-PG by moving the phosphate group over onto the number 2 carbon, and also the number 5 carbon. Step 9. Enolase removes a water molecule from 2-PG and makes some carbon-carbon double bonds, giving us 2 molecules of phosphoenol pyruvic acid. Step 10. One final step in glycolysis. The enzyme pyruvate kinase transfers the last 2 high-energy phosphate groups onto ADP through substrate-level phosphorylation, making our 3rd and 4th molecules of ATP and leaving us with 2 molecules of pyruvic acid. And really, at physiological pH, it's not pyruvic acid, it's pyruvate ions. Let's finally show them as ions. That's it, we're done with glucose. In the investment phase, we started with glucose. We spent 2 molecules of ATP to cram some more chemical bond energy in there. And then we chopped it in half to make 2 molecules of glyceraldehyde 3-phosphate. In the payoff phase, we stick on 2 phosphate groups that were just floating around in the cytosol, take out 2 pairs of high-energy electrons, and some substrate-level phosphorylation to make 4 molecules of ATP. Spend 2 ATPs, make 4 ATPs, profit 2 ATPs. We're left with 2 pyruvate ions, which are ready to head off to the pyruvate dehydrogenase complex in the mitochondrial matrix, and we have 2 pairs of high-energy electrons on NADH plus H+, which may or may not get to the electron transport chain. We'll have to see. If you have room in your brain for one more horrible detail, those 2 water molecules we took out in step 9, those are what are left over from the phosphate groups we added in step 6, because in step 6 I showed them coming to the party as H3PO4, but what got added to each ADP during substrate-level phosphorylation was 1 hydrogen, 1 phosphorus, and 3 oxygens. So the difference between H3PO4 and HPO3 is the water molecule that went away during step 9. Okay, that's definitely enough for now. In the next level 3 video, we'll talk some more about horrible details for the citric acid cycle. In the meantime, thanks for watching. I'll talk to you then.